Welcome to another new session of Sulagna'sBioBlog. Today I will discuss about pancreatic function test. First I will discuss what is pancreas. The pancreas is a gland that serves important functions related to digestion and the production of certain hormones. The pancreas is located within the upper abdominal cavity in close proximity to the stomach, liver and the small intestine. The pancreas is composed of two parts or lobes. The right lobe is located near the descending part of the duodenum, which is the first segment of the small intestine, and the left lobe of the pancreas lies next to the stomach. The main functions of pancreas. The main functions of pancreas are: the hormonal function includes production of insulin, which is responsible for carbohydrate metabolism, production of glucagon, which have its activity confined to liver glycogen, and the exocrine function. So the first I have discussed about the hormonal function or endocrine function, and then about the exocrine function. Pancreas is known as the mixed gland because it serves as both endocrine and exocrine glands. Endocrine means which is involved with hormone secretion, and exocrine means which is involved with enzyme secretion, when the secretion is secreted out by a duct. And for the endocrine function, when the secretion comes out and it directly mixes with the blood. Exocrine function, which is responsible for the production of digestive enzymes such as amylase, trypsin, lipase, etc. Pancreatic juice is responsible for digestion of protein, carbohydrate and fats. The common diseases of the pancreas. The first one is pancreatitis. One of the most important and potentially dangerous disease is inflammation of pancreas. It is a common disease of dogs and arises when foods high with fat content are eaten. The general symptoms for pancreatitis are chronic abdominal pain, back pain, nausea, vomiting, unintended weight loss. The next disease of pancreas is diabetes mellitus. It is the most severe and most common disease. A serious disease of pancreas is diabetes mellitus. It represents underproduction or low secretion of the hormone insulin. Insulin helps to metabolize carbohydrate taken in the diet. When inadequate or low production of insulin occurs, it causes blood sugar to become very high. As the carbohydrate sugar taken cannot be metabolized, circulating levels of glucose is high in blood. Many cells cannot use it for energy and serious side effects develop. The third disease is pancreatic cyst and abscesses. Pancreatic abscesses are small pockets of infection that may develop as a complication of pancreatitis. Next is exocrine pancreatic insufficiency. A condition represents a deficiency in digestive pancreatic enzymes and leads to an inability to digest food properly. Because the enzymes are not secreted, that's why the food digestion is not proper. So now we will discuss about the tests. Which tests are to be done for checking the action or the function of the pancreas. It includes microscopic examination of feces for number 1, detection of fat or iron digested protein or starch. Second, examination of feces for presence of trypsin. Third, serum lipase determination. Next one, serum amylase determination. Fifth one is blood glucose or glucose tolerance test. And the last one is absorption test. So the first we will examine the feces for the detection of fat. How we will do it? Fat appear in feces in the form of neutral fat globules, which can be detected microscopically by stain the diluted fecal sample with Sudan 3. Detection of fatty acids. Fatty acids can be detected by heating a mixture of fecal smear with 4 drops of 36% acetic acid. Detection of undigested starch. Starch can be detected by staining the fecal smear with 2% tincture iodine. Undigested starch will appear blue color. Detection of protein. Under microscopic examination, muscle fibers appear light yellow. Then examination of fishes for presence of trypsin. And the test is gelatin tube test. Add 5 ml of test. Add 9 ml of water to few fishes. Warm 2 ml of 7.5% gelatin solution to 37 degrees Celsius. Next, add 1 ml of 5% sodium bicarbonate and 1 ml of the fecal dilution and mix. Next, prepare a blank by placing 2 ml of gelatin and 1 ml of 5% sodium bicarbonate and 1 ml of distilled water in a second tube. Next, incubate both tubes at 37 degrees Celsius for 1 hour. Next, refrigerate the tubes for 20 minutes and read the results. Failure of sample to solidify gelatin indicate presence of trypsin. Next is film test. Add few fishes to 9 ml of 5% sodium bicarbonate. Place a drop of dilute fishes on a piece of extra film or place a strip of film in the diluted fishes. Incubate at room temperature for 2.5 hours and at 37 degrees Celsius for 1 hour. Then rinse off both pieces of film. Clear of the test film where it has been soaked with the fecal mixture. Indicates the presence of trypsin and functional pancreas. Next, serum lipase determination. Serum lipase activity is determined by colorimetric assay. First, serum is dissolved in 50 millimolar per liter trace HCl buffer at pH 7.7. Second, mix vigorously for 1 minute to obtain transparent reversed micellar solution and pre-incubate the reversed micellar solution at 37 degree with magnetic starter at 500 rpm. Third is initiate the enzymatic reaction by adding olive oil and collect each aliquot of the reactant at predetermined interval. Fourth, dilute the collected sample with 4.6 ml of benzene and isooctane and mix vigorously for 30 seconds. Then add 1 ml of cupric acetate reagent and 2 ml of cupric acetate acetonitrile mixture into the diluted sample and centrifuge at 3000 G for orthogravitational unit for 5 minutes after vortex mixing for 30 seconds. Next, measure the absorbance of colorized upper layer at 750 nanometer. Elevated level occurs in acute pancreatitis, renal failure, hepatic disease, pancreatic neoplasia and abscession. Then serum amylase determination. The colorimetric assay is done by using 2-chloro-4-nitrophenyl maltotrioside CNP-G3 substrate, which reacts with amylase and release 2-chloro-4-nitrophenol CNP. The increase in the absorbance is measured at 405 nanometer and at 37 degrees Celsius is proportionate to the enzyme activity in the sample. Elevated amylase level occurs in acute pancreatitis, renal failure, glucocorticoid administration, pancreatic neoplasia and abscession, intestinal obstruction. Secretin stimulation test. Small intestine produces the hormone secretin when partially digested food arrives into small intestine from stomach. Secretin makes the pancreas to release a fluid that contains inactive digestive enzyme and bicarbonate. This fluid moves from pancreas into the upper portion of small intestine, duodenum. This fluid neutralizes stomach acid to activate pancreatic enzymes. Procedure. A tube is inserted through the nose or throat into the stomach or small intestine. Secretin intravenous injection is given to the patient. After 1 or 2 hours, samples of fluid are taken from the small intestine through the tube and sent to the laboratory for test. Laboratory analysis. Radioimmunoassay is done to assay secretin. How it is done? Now, first venous blood is permitted to clot at 4 degrees Celsius. The serum is separated and assayed or stored at minus 20 degrees Celsius. Third, it is a two-stage procedure using a double antibody method for separation of antibody bound from antibody-free I-125 labeled secretin. Next, dilution of the assay reagent is prepared in PBS, phosphate buffered saline containing 0.25% BSA, bovine serum albumin. Next, after 72 hours incubation at 4 degrees Celsius, the soluble secretin antibody complex was precipitated by addition of goat antiserum. Sixth, after centrifugation at 2000 G at 4 degrees Celsius for 30 minutes, the precipitate and supernatant were counted with a gamma spectrophotometer. Next, blood glucose and glucose tolerance test. These two tests are performed to check the functionality of pancreas, as the hormone insulin secreted by the pancreas plays an important role in the breakdown of glucose. The next test is dual level Shilling test. How this is done: oral radioactive vitamin B12 is given, and along with it, intramuscular injection of non-radioactive vitamin B12 is also given to saturate B12 binding protein and to flush out cobalt 58 radioactive vitamin B12. The urine is collected for 24 hours for permitting urinary secretion of absorbed radioactive vitamin B12. Normal person will excrete greater than 10% of oral dose, and if less than 10%, repeat the test with addition of oral folic acid IF. If now normal, after giving the second dose, if now normal, the diagnosis is pernicious anemia or gastrectomy. If still abnormal after the second dose, if still abnormal, then the lesion must be in the terminal ileum or bacterial overgrowth. The test is done for pernicious anemia, atrophy of gastric parietal cells, lead to absence of gastric acid and intrinsic factor secretion. Second, chronic pancreatitis, deficiency of pancreatic protease to split the cobalamin R binder complex. And the third is achlorhydria, absence of another factor secreted with acid that is responsible for splitting cobalamin from the protein in food. It may be bacterial overgrowth syndrome, bacterial utilization of cobalamin, and the next one is ileal dysfunction, impaired cobalamin intrinsic factor uptake. Fecal elastase test. Pancreatic elastase appears to be more stable than chymotrypsin during intestinal transit. Exogenous enzymes do not interfere with the elastase test as they do with the chymotrypsin assay. Fecal elastase test. Pancreatic elastase appears to be more stable than chymotrypsin during intestinal transit. Exogenous enzymes do not interfere with the elastase test as they do with the chymotrypsin assay. This is all about pancreatic function test. Thank you. This is end of the today's video.